One of the big activities in the Biodiversity Genomics Europe project is DNA barcoding. And here at Naturalis, we are involved in quite a few steps in that stream. So for DNA barcoding, we are building up a reference library against which we can then determine the contents of mixed samples. So let's say a water sample, you want to know which species are in it. Well, you do the identification at a molecular level by comparing the genetic variation that you see in the sample against the reference library. And building up that reference library is what we are heavily involved in a whole chain that starts at selecting which museum specimens we want to sequence, then doing the sequencing, then with the data that we get out of that, clean up that data, and then deposit it in a database whose contents can be used downstream for these identification workflows. Now, the part where we built that database, that is the part that I lead in the project, and what we're working on is basically deploying in Europe a mirror of this system called BOLD, Barcode of Life Data Systems, which is basically a product developed by our colleagues in Canada at the Center for Biodiversity Genomics, and it has been around for many years. It is now at version 4, and what we are seeking to accomplish inside the project is to deploy version 5, which will have more functionality for downstream analysis, more kind of high throughput, and the technology underneath it is totally different, maybe not that interesting, although I could speak about it at length. But anyway, so what we're working on is deploying here a system that kind of exists now in a beta version. And that's something that we have just accomplished. So actually last week there was a little bit of celebration because this early version is now hosted in Europe. And that sets us up for further development. So in the coming months and years, we are going to further refine that system, make it perform better, and tailor it more for our needs within the project. So there's a lot of also communicating with the different stakeholders to make sure that this system does what they need, so that we can effectively use it in what we're trying to accomplish in the end, which is to measure change in biodiversity at the molecular level with reference to these existing data systems.